All right, guys, this video is for guys who are not in class or fourth period. I was not there. Mr. Clark, obviously, as you can tell, is here. Hi, Mr. Clark. So hopefully this helps you. This video is just going to go over about DNA replication. The thing with DNA replication is there's a lot of enzymes that you need to know, and you got to be able to figure out what they do. On the left side of the board, you can actually see a list of those 8 enzymes, what they do and what their function. I'll put that at the end of this video as well. Between the notes here, you're going to read an outline, 12.1 that's going to have it. I got a homework assignment that's tied into that Mr. Clark's going to hand out and explain. And you've got an activity. All of those things are basically just covering DNA replication 4 different ways. It's important to know the parts, what they do, how they function. And so carrying that into what we talked about last time, we talked about DNA being a double helix. And one of the things that was important in, Watson and Crick's work was they said, in that article, yeah, it's a double helix, but it also lends itself to how it could replicate. And we mentioned that last time. If you think about the process of it, it's really not that challenging. What would you have to do? Well, you take that DNA molecule, you'd open it up. You'd have to find some system to open it, split it open, figure that out. Once you were able to do that, you'd have to hold it open, keep it apart, keep those hydrogen bonds apart, and then you'd have another something come in and start adding bases following the base pairing rules. And based on the fact that DNA had those base pairing rules, it was important, it's easy to see how that naturally would be added on to the new segments, as you can kind of see in the diagram here. So open it up, start adding the bases, and then we're going to get 2 new strands that are complementary off of that parental strand. The last thing that's really important to note, again from Watson and Crick, was the fact that the structure of DNA runs anti-parallel, which just simply means if one strand runs in this direction, the other strand runs in the opposite direction. They're not running in the same direction. So that term anti-parallel is going to be important for understanding how DNA replicates. When we look at the structure of DNA and how it processes and makes new DNA, the first thing has to happen is the enzyme needs to be unwound. So as you look up on the board, you can see the first enzyme that we're going to mention, enzyme number 1, is helicase. And helicase's job is to open up the enzyme. It opens the helix. It allows it to unwind, and it helps it from allowing those hydrogen bonds to open. As those 2 strands separate, we got to be able to hold them apart. That's where our next one, number 2 on the board, comes in. That's the SSBPs. SSBPs stand for single-stranded binding proteins. Their job is to keep the helicase open. If you think back, those hydrogen bonds that the helix opened will just form right back together as the helicases move through. So the SSBPs come in, and then their job is to hold open the molecule. The way I always think about it is if you're ever on a boat, you see how the boat actually cuts through the water? The water spreads open, and then right behind the boat it comes back together. Well, the SSBP's job is to keep that water from coming back together in the form of hydrogen bonds. Again, called SSBPs. Their job is to hold open and prevent it from coming back together. The point at which the actual DNA molecule opens up is called the replication fork or bubble. So you can kind of see it here. I have it drawn up on the board as well. But when you actually zoom in into it more, we call it a bubble because wherever I opened it up, let's say I open it at this location, it's going to open up there as well. So it forms more of a bubble versus an actual just one fork. For our example and what's on the board, it's this section right there. And you can kind of see the next enzyme that's going to be really important is going to be number 3, called primase. Primase, its job is to add the primer. Its job is to add the primer, and the way you think about that is when I were to go to paint something, if I was going to paint this cement brick in the room and it doesn't have any paint on it, you need to add a primer. The primer sticks to the cement, and the paint sticks to the primer. The next molecule that's going to actually be adding the bases can attach and doesn't know where to attach to the DNA template. And so the primer attaches and then signals our next molecule, DNA polymerase, to attach at the right location. So again, it's an important thing to think about is that the next enzyme is called primase, and it lays down a primer. A really important stat or thing to remember with the primer when it's added is that it's made out of RNA, okay, not DNA. Just keep that in the back of your mind because, again, we're making DNA, so we're adding this primer so that it attaches. Eventually, before it's all said and done, we got to remove it and replace it with DNA. But that's for a little bit later in the notes. Right now, the primer is the next molecule. It attaches to the DNA strand, and allows the next molecule to do its job. That next molecule is called DNA polymerase, and there's actually 3 of them that we're going to talk about. The main one, the most important one, and this is why I have asterisks up on the board for it, is DNA polymerase 3. DNA polymerase 3 does a couple things. It adds bases, it attaches to the primer, and adds bases following the base pairing rules. It also has the ability to backtrack, proofread, find mistakes that are occurring in the molecule. Even with that proofreading, errors do occur. They happen 1 in a billion nucleotides. That may seem like very little, but you think about how many replications actually take place in the human body in a given second. 1 in a billion is really not that great of a number. And so there's going to be other checks that we talked about in mitosis, and we'll talk about some other ones in class next time to help prevent some of those mistakes from occurring. DNA polymerase 3, as I mentioned, if I had to name the number 1 molecule in this process that does the most things, it's DNA polymerase 3. Here's a slide for our video, which talks about our next enzyme, which is topoisomerase. I'm not going to show this video now. You can watch that for homework. It's listed on Canvas in the 12.3 folder that's called, and this one again is called the twisting. This is an important issue here. If you could think about rope twisting, and you were to untwist it, so you were to pull, these strands here apart, that as you pulled them apart, in this region up here, it would get tighter together. And so as we look at the molecule helicases unzipping this molecule here, it's going to create tension up ahead. And so topoisomerase is going to come in and relieve some of that tension. So that's the second, or the next enzyme that we need to know. It's list number 7 on the board. So make sure you know that video that corresponds that. It's on Canvas. Make sure you're able to watch that. An important and complicated thing that I can't emphasize this enough, I can't highlight this enough, it's on the board. Make sure you understand this. Topoisomerase, just like us, we have to read in a general direction. In other words, most of us read left to right, top to bottom. Topoisomerase has to read the template strand. It reads in the 3' to 5' direction, and it's going to add new bases on the new strand, so it adds the bases in the 5' to 3' direction. This right here is incredibly important and one of the easily mistaken things on our test, so make sure you know that. And because it does this, it creates these strands in different ways. What it ends up doing is it creates what we call a leading strand and a lagging strand. The leading strand is not necessarily the best name. It's more of a continuous strand happening in one direction, one continuous flow. So in the diagram I have on the board, the top is considered the leading strand. The template strand is reading it in the 3' to 5' direction. Here's the 3, there's the 5, it attaches, and it's going continuously in that one direction, making one continuous strand. But on the bottom, as the strand opens, it'll open here, make a strand, helicase opens it up more, goes here, helicase moves a little bit further down, creates another strand. The bottom strand, the lagging strand, is built in segments. Okay? Each one requires the addition of a primer. So each one of these has a primer. Up here, we only needed one primer, and that primer is laid down by the enzyme primase, as we mentioned earlier. The next enzyme that's going to play an important role is DNA polymerase 2. It's up on the board as number 5. Its job is to proofread and edit, and to make sure there's no other mistakes that may occur. The last one that's really significant is the enzyme DNA polymerase 1. What it does is it'll find these areas of primer. Remember, these primer, as I mentioned earlier, are made out of RNA. What this will do is remove and erase that primer. It's going to erase those primer and fill that in with the corresponding DNA molecules. Notice we still have segments. They're not officially attached. And so in order to fix that is our last enzyme, ligase. Ligase job is to come in, get rid of it being separate units, and it fills in the blanks to make it one continuous stretch. It fixes the backbone. That's the last enzyme. It ties the backbone together. DNA polymerase 1 removes and replaces the RNA primer, and it will do that anywhere the primer is. So it'll do it once on the leading strand where the primer is laid down, and all the different beginning of the Okazaki fragments on the lagging strand. Again, those Okazaki fragments are the small segments found on the lagging strand, and this will make a little bit more sense when you watch those videos that I have for you. Ligase, as I mentioned, is the last enzyme, and it will wind it back. Here are the videos that I told you. This one is the first one with the enzymes. Go ahead, and what I'm going to have you do now is actually watch those 4 videos that are posted on there. You can go ahead and get your headphones out, watch those 4 videos in order, starting at the top, going down. If you don't have your headsets, maybe a partner next to you. I don't want too much noise here. Should only take a couple minutes. When you're done, Mr. Clark, you let Mr. Clark know and he'll restart this video. So I'm going to stop the video now, and or Mr. Clark, go ahead and stop it now. Watch those videos that I have on Canvas, and then when you're done, we'll resume the video. DNA replication occurs with multiple replication bubbles. I'm not going to get too much into this. Why does it do that? Well, it's like if I asked you to copy down the biology textbook, it would take you forever. But if I asked every student at prep to copy down 1 page, it'd happen much quicker. Why do we do it as quick as possible? The DNA is not exposed, therefore less damage can occur, and it helps protect it. This diagram basically shows summary of DNA replication. I think something like this on the test would be really important for you to see and understand. What I would do is have it on here, and I would have stuff like you'd have to label the numbers, you have to label an Okazaki fragment, RNA primer, primase, DNA polymerase, DNA ligase. I would also have on here somewhere topoisomerase, which isn't represented. I'd probably have to add it in. And you'd have to know the difference between DNA polymerase 1 and DNA polymerase 3. I will not have you identify DNA polymerase 2 on the test.